Hello everyone, today we are going to see what is DNA sequencing. So the objective of this particular video is to understand what is DNA sequencing, what is the history, and what are the basic methods of DNA sequencing, that is chain termination and chemical modification methods. So to start with, when we talk about DNA sequencing, it is precisely nothing but to understand to determine the order of the nucleotides in the DNA. We all know that you have 4 different types of nucleotides which are randomly ordered in a DNA, so you have to know what is the sequence. So we need to determine the order of nucleotide bases in a strand of DNA for sequencing. So what is the need for this DNA sequencing? We require it for characterization, for any forensic determinations, then molecular archaeology, gene-to-gene interactions can be studied, gene-protein interactions can be studied, and very important, in cloning also we require the sequences of a particular gene. So when we talk about the methods of sequencing, so there are basically, when we talk about the conventional methods, there are basically 2 conventional methods. You have Maxim and Gilbert method, which is a chemical sequencing method, and you have Sanger's method, which is a chain termination method. So both robotic and automated sequencing, which is a norm today, are based on these methods. So we'll start with the Maxim and Gilbert method. So in 1976 and 1977, Alan Maxim and Walter Gilbert, they developed this DNA sequencing method, which was based on chemical modification of the DNA. And subsequently, whatever the modified bases were there, they were cleaved off. So we cannot use this sequence later on. That is a fact. So chemical modification of DNA, you have radioactively labeled DNA, which is labeled using by a kinase reaction using your p32 ATP, then purifying this DNA fragments which are to be used for the sequencing method, and then the chemical treatment is done to break open this DNA, and they are run on the gel to determine what is the sequence. So you have the ploy nucleotide kinase activity. We can see this is our sequence, and the radioactive ATP, the phosphate 32 phosphate, is labeled with this particular sequence. And then we start with the chemical modifications. Now there are different types of chemical modifications that can be seen. So first, when we are modifying with dimethyl phosphate. So DMSO only alone can modify only guanine, and DMSO along with formic acid can modify guanine as well as adenine. And then using piperidine, this sugar phosphate backbone could be broken off. Then when we are using your hydrazine, so hydrazine alone can react with cytosine as well as thymidine, whereas hydrazine along with NaCl can modify only cytosine. And then again piperidine could be used to cleave the sugar phosphate backbone. So then we can have then 4 different types of reactions as we saw. So one is only with your DMS, and then one is only with your hydrazine, one would be hydrazine with salt and hydrazine with the acid. So you can see here you have only the guanine, the ends only the specific sequences which are ending with guanine. Here you can see which is ending with guanine as well as adenine, here with cytosine and thymine, and here you can see only cytosine. So then when we run them on the gel, you can have all these 4 reactions. So the contents of every reaction would be then loaded onto a single well on a agar gel electrophoresis. So you can see this is only the sequences which are ending with guanine. These are the sequences which are ending with guanine as well as adenine. So that is the reason why you will see that here you can see the bands which are there in this first well, the same bands would be definitely being found in the second well. Apart from that, you will have these extra bands as well. So these extra bands are for adenine, and the common ones are going to be for guanine. Similarly, here also you will find the same thing for the third and the fourth one. Because this is the combined one and this is just for cytosine, you will see that the bands which are seen in the well 4, the same bands would be seen in well 3 as well. Apart from that, here you will be seeing some extra bands, so that would be corresponding to your thymine. So that is how the sequences can be, we can obtain the sequences, or they can be read from bottom to top. Why bottom to top? Definitely because the smaller the sequences or smaller the fragments, they would be running farthest. So that is the reason why we have to read it from bottom to top, and that is how we can determine the sequence of a particular DNA. So this is the summary of what we have already seen. So we have to first label our DNA and then aliquot it in small 4 tubes. As I said, 4 tubes because each tube will correspond to the modification reaction, the 4 different types of modification reaction that we have already seen. So once they have been modified by the chemical reactions, then they would be cleaved, and then the gel electrophoresis would be done and autoradiography would be obtained, from which we can interpret the results. So advantage of this particular method: no premature termination due to the DNA sequencing, because here we are totally dependent upon just the chemical reactions. So when we are talking about any kind of your biological reactions or depending upon some enzymes, there are chances that premature termination will take place, but here those terminations will not take place. So stretches of DNA can be sequenced, which cannot be done by enzymatic method, that is your Sanger's and Gilbert method, the Sanger's method, which we would be seeing in the next video. Purified DNA can be read directly. Homopolymeric DNA runs are sequenced as efficiently as heterogeneous DNA sequences and can be used to analyze DNA protein interactions, which is called as your footprinting, and can be used to analyze the nucleic acid structure and epigenetic modifications. Okay, epigenetic modifications are like when because of some environmental changes you have addition of methyl groups, or these are just an example, some other modifications that take place in the DNA that can also be analyzed using this particular method. Talking about the disadvantage, not very widely used. The reason because radioactivity is used in this particular method and the chemicals that we are using are toxic. So you require very trained experts to do this particular method. It requires extensive use of hazardous chemicals. It has a relatively complex setup and technical complexity is involved. It is difficult to scale up. That means at a larger scale we cannot perform this particular method. It cannot be used to analyze more than 500 base pairs, and the read length decrease from incomplete cleavage reactions. And the most important thing, nowadays we are more dependent upon DNA kit methods, but because of the nature of the chemicals which are used in this particular method, it is difficult to make such kind of your sequencing kits using your Maxim-Gilbert method. Thank you. In the next video, we will be seeing the Sanger's method, which is an enzymatic method. Thank you.